Well, welcome everyone to today's Safer World by Design webinar. As you almost certainly realize, new approach methods, their development in research and their translation into practice is one of our main interests in our community. And for that to be successful, a lot of aspects need to be taken into account, for example, in terms of good practices, in vitro, in silico, good cell culture practice, and so forth. And today's talk by Sandra Koek at the Joint Research Center of the European Commission will, I think, give a quite sweeping overview of a lot of great work that's been going on by that group and with a lot of collaboration to help bring these new approach methods towards acceptance and more regular use as tools. So with that, I hand over to you, Sandra, and please go ahead.
Okay, thank you so much for inviting me for this webinar. And I would like to go to the first slide. Next one, next slide. So I give you a little bit an idea what I will be talking about. So I will be starting with an introduction. And then I will go and give you an overview of 2 decades, almost 3, I would say, of ADME in vitro and in silico dynamics and kinetics methods for regulatory purposes. And then in the next part of my webinar, I will give you an idea why when we are talking about new approach methods, especially for their application in regulatory testing, why we need a framework and what kind of activities we need to characterize these in vitro tools for use for specific decision making. In that part, I will also elaborate a little bit on the issue of species difference in metabolism. And then I will proceed with an example why whatever we do for, let's say, global comparison of method data of these new approach methods, why globally there is some kind of need for standardization and harmonization of in vitro metabolism methods. And to show you that, I will show one of the examples of a validation study we have been doing with in vitro metabolism methods, the CIP induction validation study, which gives you a good flavor of what I'm trying to transmit to you all. And then I will end with a last example, giving you an idea what might be current regulatory needs, immediate current regulatory needs, where we badly need all these new approach methods, as Barry was indicating, and also where we need then in vitro metabolism methods. And I will give you an example. So let's go to the next slide. So obviously, ADME has as a key component a metabolism, and definitely it's also a key component in many integrated strategies. So when we think about metabolism, we have many aspects that we need to deal with, understanding, let's say, several information needs that are given by these methods. So we basically need to know when a chemical is entering in the human body. The first thing is that we need to identify important metabolic pathways, often in different species, because obviously in toxicology we still have the target species that are still used. We need to provide knowledge of the specific human enzyme isoforms involved in metabolism. Also, we need some information on metabolic stability. We need to have information on the type and structure of the metabolism of the metabolites, and also any metabolism-mediated toxic effects. And also, and this I will show you a little bit more in depth, we need also information via mechanistic methods to see if these compounds can induce or inhibit biotransformation enzymes. And so for all these aspects, during my presentation, I will give you some examples. Working at the European Commission Joint Research Center and at my, let's say, organization, the European Union Reference Laboratory for Alternatives to Animal Testing, since I came there, obviously, one focus of our work is to work on these new approach methods and to see that we can use them in a reproducible, transferable manner, and that obviously whatever we use, we need methods that are relevant and that from a mechanistic point of view are predictive. So next slide. So I introduced that we are working quite a long time already, and it's almost 3 decades. And I'm showing this slide, and Christoph Chesne, that is also on board today, he will remember this slide because we were there together in Marseille in April 1992. And these were, let's say, the first series of experiments where we tried to isolate reliably and efficiently human hepatocytes. Now, as you can see from the picture, next, you see that there was some standardization still needed, but that's how it all started. So next slide. When you see then 15 years later from these experiments, next, a lot of work has been done with a lot of experts to basically get more insight in how we can use practically hepatocyte cultures in routine testing. That was actually one of the first workshops that was also organized by ECVAM. And over the years, you can see it on the timeline, many experts in the field have been brought together to look a little bit more into depth on the long-term use of hepatocytes cultures. Also, they looked into the issue of metabolism-mediated toxicity as a bottleneck. And obviously, we looked at dynamics and very soon also to kinetics. And it is clear that in pharmaceutical industry, where I worked in the beginning of my career, they definitely dealt already very early on with pharmacokinetics in drug research. And obviously, this is an area that you also have to deal with when you do chemical risk assessment. So I provide these references, and then probably after the webinar, we will provide them as a list. So quite a lot of work has been done. And the next slide shows that another 10 years later, a lot of work again has been done by many experts to look at the possibilities of using metabolism and kinetics, new approach methods for regulatory decision making, also in fields different than drug testing. And I think we can go on for the next decades to publish on these methods. But what I think is now important, and this is something also what you feel now, that finally we need to really start implementing these new approach methods for regulatory decision making. And therefore, we need some kind of a framework also at regulatory level, at global level, where we agree how to use these tools in combination, as has been done already for many years in pharmaceutical industry. Next slide. So during the work, let's say OECD has been a very important player now to start also moving some of these new approach methods like PBPK models into the global regulatory arena. And this is an example of a guidance document that is being drafted by my colleague Alicia Paini with others to bring, let's say, the use of PBPK models globally at the forefront of the attention. So this guidance document will be published soon. Next slide. So also at European level, the European partnerships on alternatives to animal testing already since the beginning has been a very big ambassador of using these new approach methods. And also today, still there are projects that are being supported to support the application of physiologically based kinetic modeling in safety assessment, and also some open source tool to do quantitative in vitro to in vivo extrapolation, which is obviously an important element in this story. And this is one of the projects that is being led by Joyce Loisy, who has been also a very great ambassador towards these open source tools. Next slide. So going to the third point of this webinar, as I said in the beginning, we can publish about these methods, we can come together, discuss what has to be done, but now definitely we need a clear, globally agreed framework, how we use these methods, and also to agree how we should characterize these in vitro methods, metabolism methods, to have also trust from the users and also from the regulatory decision makers to use them efficiently. So I will show you some issues where we as an organization contributed to strive for harmonization of the data that are coming from these in vitro metabolism methods. I will start with the example of metabolic stability clearance. So you can go to the next slide. So basically, metabolic stability and metabolite identification is quite important. And as I said, in toxicity testing, we still use animals. And one of the issues to deal with is understand, obviously, if there are differences between the different toxicity species and human and let's say the human metabolism. So one of the early publications we did in 2009 was actually seeing what are the quantitative and qualitative differences when you are looking at metabolic stability data and metabolite identification between, for instance, human and rat, homogenate and microsomes. So in that case, we used subcellular fractions to do that. And this work is currently being followed up, let's say, with our colleagues at EFSA, where we are working with different experts in a group looking at comparative metabolism to see what are the differences between the species in terms of the metabolites formed, if there are unique metabolites, if there are disproportionate metabolites. And the test systems in that time of the publication, we used microsomes and homogenates, but obviously you can now reliably use human hepatocytes, and also there has been work ongoing to look at tools like the HepaRG test system to support some of the work in that field. Next slide. So one of the things we started with talking about the framework was a first, let's say, mini framework to characterize in vitro methods for human hepatic metabolic clearance. Next slide. What we saw immediately when you deal with clearance data, that around the globe, people are using different test systems, different test system configurations, and also there is a variability in the measured parameters when they do these studies. And this you can see here on the slide, and this is also published in the publication that I just have been showing. Next slide. Another thing that we learned, and this is quite interesting for you to look at carefully, is when we look at data generated on human hepatic intrinsic clearance. And this is an example where I give information on using human hepatocytes and red hepatocytes. On the left part of the slide, you see on the bottom, you see 37 chemicals from 30 publications. And on the right slide, right part of the slide, you see red data on 10 chemicals from 15 publications. And you see definitely that there are, as we saw already in the previous slide, large variations in the protocols. But the fact that there are these large variations in the protocols, you see also that for the same chemicals, the intrinsic clearance data are orders of magnitude different, because you see expression in a log scale. So that's quite worrying. When you work, obviously, in your organization and you do intrinsic clearance data on these chemicals, you can compare within your company or within your organization. But if you want to compare absolute values between different players globally, there are quite some differences in the data. The question is, how important are these differences? And do they, for instance, have effect when we use them as input parameters in PBPK models? Next slide. So when we look to the variables that are driving these differences in absolute in absolute manner, you see that the variable importance is depicted here in this slide. And obviously, variable importance can be driven by the medium, by the use of serum, and many other factors. So there is something that is clearly methodologically that is driving this variability. So for all the details, you can have a look into this publication where we elaborated a little bit on that. So next slide. So after this, obviously, if you want to use a method in this toolbox for regulatory decision making, some kind of standardization, as I said in the beginning, might be needed, especially when you work with some quantitative methods and you want to compare also absolute values that are obtained with these new approach methods. So in this part of the presentation, I will give you an example that it is possible to standardize these methods, because that is, let's say, our core business. And as an example, I will give to you a validation study where we looked at phase 1 biotransformation induction. So in this case, it was CYP induction. And I show this example because it gives you a flavor of what can be done with this type of approaches, also to see that this can be applied to other new approach methods that we badly need in our metabolic toolbox. Next. So here you see a little bit a background on CYP induction. So basically, we know in the area of endocrine disruptors, which is an area for us at the Commission that is quite important because we have our community strategy on endocrine disruptors. You see that for endocrine acting chemicals, this underlying mechanism like CAR and PXR are playing extremely important role in this area, not only for phase 1 biotransformation enzymes, but also for phase 2 biotransformation enzymes. Next. So in one of the big validation studies we are running now, which is the thyroid validation study, I give you some example. We are also looking to inhibition induction of UDP GTs and sulfotransferases. And in this area, as you know, also phase 3 is important. So we also need to think about transporters in our toolbox. Next slide. So when we go to the underlying molecular mechanism of these methods, these metabolism methods, as we say, well, they are very important because obviously for many of the chemical entities we are dealing in our different, let's say, industries, you see that many of the compounds that are listed here have a link with this PXR and CAR activation in the field of endocrine disruptors. So here I just listed steroids, pesticides, phthalates, alkylphenol, and PCBs. But obviously there is a lot of evidence in literature on the understanding of the underlying mechanism. Next slide. What is also interesting there, obviously, is the species differences. So you see also with several of these compounds that there are species differences between, for instance, PXR and CAR. So here you have an example. Rifampicin is a potent activator of human PXR, but not of mouse PXR, etc. So you have a lot of examples here where you see that we also, when we are interpreting the data coming off these methods, that it is important to go to the mechanistic underlying principles of the different methods and see that you have in your toolbox all the methods together. Next slide. So when we look at this validation study that we did for this human CYP induction validation study, how we approached this, we used two different test systems, which were HepaRG cells and primary human hepatocytes. We brought together 3 independent laboratories for each of the test system and the related CYP induction methods. And we also had a group of experts that were overviewing the validation study. And basically, all the labs work together to get, let's say, the methodological aspects of the protocols that were initially given to us much more aligned between all parties. Next slide. So within the area of ATME in general, most of these new approach methods, they use a part which is cell culture based and a part of analytics. So when you do this validation study, obviously you need to do a part standardization on the cell culture part and a part standardization on the analytics. Here on the slide, you see the underlying principle of the CYP induction method. We have been looking at 3 CYPs for this induction method, 1A2, 2B6, and 3A4. We have been using reference items and specific enzymatic probe substrates, and then also measuring by LC-MS the metabolites that were formed, and we used an N-in-1 approach to do so. Next slide. So all the details on this validation study can be found in this publication, but basically we found a very good within lab reproducibility and also very good between lab reproducibility. Next slide. So concordance in terms of predictivity was also very good. For the HepaRG cells, we had a very good, let's say, performance for all the test items that we selected. In this particular validation study, we have been using human in vivo data because obviously, thanks to the collaboration with pharmaceutical industry, we had background human in vivo data for all these compounds listed. So this was a validation study where we could compare our human in vitro test systems and methods with really real world data from the clinical studies that were done on the compounds listed. So most of, let's say, the induction data that we got were correctly classified, but as you can see here on the slide, it is not a black and white exercise. You need definitely to understand that you need to reason when you see this data to make a correct decision on the validity of this surrogate test method in confrontation with the reality, which is induction in the human body, where you definitely have also differences between different human beings. Next slide. So in the last part of this webinar, I would like to show you why we need all this let's say, work quite urgently, because we are dealing with complex questions in toxicology, and there is a need, let's say, for new approach methods in different areas. One of them, as I indicated already during my presentation, is the area of endocrine disruptors. So we have now a new OECD test guideline for most of the endocrine related effects, so be it estrogen, androgen, steroidogenesis, but also we need thyroid disrupting information. And so one of the areas that we have been taken on board is a large-scale collaborative study to tackle globally the thyroid disruption health burden using a combination of many mechanistic methods. So for doing that, we're following up a little bit the work in the Commission on that area. Next. But we also have seen appearing guidance documents from our European agency where we need to assess the potential of thyroid disruption. So we need definitely a battery of methods. Next. And also OECD already in 2014 made a very nice scoping document on what kind of new approach methods in this area are needed to further the field. Next, and obviously we worked together with OECD to have also a guidance document on good in vitro method practices. So with this validation study, we're basically applying that guidance document to completely to run this validation study. Next slide. So this is the validation study we are coordinating. We are using as a validation capacity the European network of validation laboratories. We have 15 EU NetVal laboratories, which you see on the right slide, right part of the slide depicted that are involved in this validation study, and on the left side are the developers of methods, mechanistic methods that are important to identify thyroid disrupting compounds. Next slide. So in this thyroid validation study, we are basically having 17 to 18 mechanistic methods that have been identified in the OECD scoping document. Next. And we try to implement in this study all the practices that we have been learned over the years in terms of validation of mechanistic methods. And also, as you can see in this project, we have also a block on metabolism and excretion. Next slide. Obviously, this is an extremely complex study with many mechanistic methods. And one of the things everybody is ambitioning that we also can start really to implement, let's say, the network approaches, AOP network approaches, to combine the information that will be gained from these studies. But obviously, it will take time, yeah, to combine all this information. And we are looking forward with many collaborators in the future to look at this data once the validation study is finished and how we can combine them in specific approaches. Next slide. So this study, as I said before, is also much welcomed as a follow-up of the ECA-EFSA guidance, and already in the appendix A of this guidance document, we see also that there is definitely need for mechanistic methods. As you can see in the guidance document, there is indicated we need methods for CAR-PXR activation, hepatic phase 1 and phase 2 induction, and then obviously methods also to deal with the specific signaling pathways that are important to identify thyroid disrupting effects. Next slide. So in the particular validation study, these are the methods currently, the mechanistic methods currently in the block of metabolism and excretion. So we have a method that is dealing with deiodinase inhibition, one on glucuronidation, inhibition and induction, and one on sulfation inhibition or induction of sulfotransferases. Currently, we are using hepatic test systems. It can be microsomes, it can be engineered cell lines. We have cryopatocytes, and obviously we're also looking into the use of HepaRG, because it was a very reliable test system in our validation study on CYP induction. Next slide. So I think a lot of what I've said before will be also further discussed in March 2021 in an SOT workshop. And today is the last day for abstract submission for SOT meeting in general. So if anybody wants, let's say, to continue the discussion on this future framework of application of in vitro metabolism and QI-VIV models to inform risk assessment, I think all the colleagues that will be presenting in that workshop are looking forward to see you there. Next slide. So I also would like, for those that are really stimulated to publish and have work done in this field, we have a new special issue on the topic. And we are looking forward to get any publications that can contribute to this framework. So please have an abstract, a look at the abstract that we are putting together for this special issue. This special issue is dedicated to a special man, next, which is Alfonso Lostia, which was a colleague of mine at ECVAM and of all of us, and he was then also working with our colleagues at EFSA. Unfortunately, he's not with us anymore, and we dedicate this special issue to him. He was one of the ambassadors at EFSA for really moving the area of toxicokinetics and metabolism forward. My colleague Alicia, with others, next, is also doing a specific issue on computational toxicology. So for those that are interested in that, have a look also in the special issues of MDPI if you want to contribute. And then last, not least, I would say, next slide, I'm just an ambassador because I have been working in this field now for many, many years. And the most of the joy of my work is because obviously all the people that are involved in this field of really trying to make a difference in this area for using efficiently these in vitro methods for regulatory decision making. So I hope that I gave you some food for thought, and I'm looking forward to any questions if I can help to answer them. Thank you so much for listening to me. And the last thing is obviously clear: collaboration is faster progress. Thank you very much.